So this slide is a repeat from the previous video, but I just want to make sure that we hit on somatic cells. I don't think it was formatted quite right. And so somatic cells are also known as body cells in an organism. They have the same kind of number of chromosomes. So regardless of it being skin, heart, or muscle, in this case there's all 46 chromosomes. Fruit flies, in their skin, heart, muscle, they all have 8 chromosomes. So interphase is a period of cell growth and development. DNA replication occurs during interphase. The cell also grows, carries out just normal cell activities. It replicates all other organelles. The cell spends most of its life in this phase. Things that can affect interphase would be nutrient supply, amino acids, if they're present or not, the temperature, room for growth. Those all can affect, when the cell's growing, replicating, and undergoing its regular day-to-day activities. Here's an example of a cell in interphase. When you look in the microscope, most of the cells will be in interphase, so they should look like your standard, typical, normal cells. There are 3 subcategories or steps of interphase. First one is G1, and this is when the cells grow in size. What happens is they synthesize messenger RNA and proteins are being made. Once growth and synthesis complete, they'll enter the next phase of the cell cycle. So the word synthesize means to create or make. Side note, in humans, the cell will be in G1 phase for a third of its total life. So this is the phase where it spends the most time. S phase. DNA instructs a cell that it must be duplicated. So it's necessary to fulfill that in the goal of the entire cycle, which is to produce 2 identical daughter cells. So if it doesn't duplicate or replicate its DNA, it cannot produce 2 identical daughter cells. So the whole goal is to create 2 identical chromosomes. This is really important to know, the goal of S phase. Side note, damage to DNA is kind of detected and fixed during this time. It's kind of like they're checking over their work when they're replicating it, and if they notice they made a mistake, DNA is pretty good about fixing that. G2, this is a period of rapid cell growth and protein synthesis. So again, we see that word synthesis, which means to make. So they're rapidly making protein and they're rapidly growing. Basically, it's readying itself for mitosis. Side note, G2 phase may not necessarily be part of the cell cycle for some cell types. They will just proceed or skip this G2 phase and go right from S phase directly into mitosis. Mitosis, this is the division of the nucleus into 2 nuclei, each with the same number of chromosomes. So it's really important to note that it is the division of the nucleus. Mitosis occurs in all somatic cells. So those are your skin cells, your brain cells, your muscle cells. So why does mitosis occur? And it really needs to occur so that each new daughter cell has a nucleus with a complete set of chromosomes. So it's really important that if this is my parent cell, you notice there's 2 blues, and each one of these daughter cells have 2 blues. And secondly, you can see there's 2 reds, and each of these daughter cells has 2 reds. You need to have identical daughter cells, and so that's why we need to have each daughter cell has to have the same complete set of nucleus and chromosomes. So again, here's just our cell cycle timeline again. Cytokinesis, this is the division of the rest of the cell, so the cytoplasm and the organelles. So mitosis, if you recall, is the division of the nucleus only. Cytokinesis is the division of the rest of the cell after the nucleus divides. So in animal cells, the cytoplasm pinches in And in plant cells, a cell plate forms. So here's my animal cell. And you'll see the cytoplasm starts to pinch in right here. And then in plant cells, you'll start to see notice this plate will start to form. And over time, it just forms more and more. After mitosis and cytokinesis, the cell returns to interphase to continue to grow and perform regular cell activities. This next slide just shows the microscopic view of an animal cell and a plant cell. So here's my animal cell right here. And you can see the cytoplasm starts to pinch in. When it pinches in, what's happening is it contracts these ring of microfilaments that cause it to pinch in. And we call this a cleavage furrow. So here's the cleavage furrow where it pinches in. In a plant cell, we'll start to see the formation, if you can see that very well, formation of this cell plate. And then eventually it forms it more and more until we have this new cell wall. So the cell plate is when it's not all the way connected. Here's they start forming the plate. And then when it's all the way connected, we call it a cell wall. So just a summary, we have interphase, then we have mitosis, then mitosis is made up of prophase, metaphase, anaphase, telophase, and then cytokinesis. This slide formatted a little bit funny. So this should be here. And then when the cells become old or damaged, they die and are replaced with new cells.